The discovery of CRISPR-Cas9 by Jennifer Doudna and Emmanuelle Charpentier in 2012 has changed the world. It has led directly to breakthroughs in medicine and agriculture that will save and improve the quality of lives of thousands. They were rightly awarded the Nobel Prize for Chemistry in 2020. Jennifer Doudna was born to a family of educators in Hawaii, and thanks to the incredible local flora and fauna, expressed an interest in biology from a young age. She went to college first in Claremont, California, before getting her PhD from Harvard. After graduation, she conducted research at both Harvard and Yale, as well as Massachusetts General Hospital. After making several discoveries and winning several prizes, she moved to her current position at Berkeley. Emmanuelle Charpentier was born and educated primarily in France. She received her bachelor's in microbiology and genetics at the University of Paris and got her PhD at the Pasteur Institute. She then conducted research in Sweden, Vienna, and New York, discovering key mechanisms in bacterial RNA and immunity. Having met at a conference in 2011, they teamed up and the very next year identified the CRISPR-Cas9 protein. This was first identified in the bacteria Streptococcus pyogenes when Dr. Charpentier discovered tracer RNA, which acts as a guide. This RNA could guide the CRISPR-Cas9 protein to a specific DNA sequence where it aligns and then cuts the DNA. New DNA can then be placed into the genome, altering it permanently, either disabling a harmful gene or repairing a helpful. This discovery is already transforming medicine. After its initial discovery, its first test was sickle cell disease. Sickle cell disease is a point mutation in the gene that codes for adult hemoglobin. Researchers targeted the BCL11A gene that controls the production of fetal hemoglobin, causing the body to produce more of it and thus eliminating the symptoms. It passed this test with flying colors, completely removing symptoms in patients. This has been replicated and confirmed, leading to the market approval of Casgevy. It is also currently being used in cancer research, both to generate new models and using gene editing to make existing drugs more effective. Scientists at the Dana-Farber Cancer Institute found that deletion of the PTPN2 gene in tumor cells made them more susceptible to PD-1 checkpoint inhibitors. PD-1 in T cells binds with PD-L1 on the surface of the cancer. This represses the action of the T cell, preventing it from destroying the cell it attaches to. In a normal body, this prevents the immune system from destroying cells that are supposed to be there. Some cancers overproduce this on their surface to act as a disguise. PD-1 inhibitors bind to this site, blockading it and allowing the T cell to do its job. PTPN2 usually puts the brakes on immune signaling pathways that would otherwise smother cancer cells. Deleting PTPN2 ramps up those immune signaling pathways, making tumor cells grow slower and die more easily under immune attack. CRISPR is also helping keep us fed. Although GMOs have been on the market since the 90s, the precision and speed enabled by CRISPR is once again revolutionizing agriculture. It took less than a decade from its inception for this tool to be used in the production of safe and legal food. In an amusing twist of fate, both the first GMO and the first CRISPR GMO to be approved were tomatoes. The new tomatoes claim to be high in gabapentin and claim that this chemical will help reduce blood pressure. While the FDA hasn't commented on the health claims, they have certified that it's safe for human consumption. Soon, new cereals and other staples will be approved to deal with threats like droughts, hotter than normal winters, and disrupted rainfall patterns. With climate change altering the very foundations of our world, CRISPR will enable us to evolve our crops fast enough to face this threat. Despite the newness of this technology, there are already laws, regulations, and policies in place to guide our research and prevent its misuse. The questions of genetic manipulation in humans was raised in the movie Gattaca in 1997. This movie, combined with the successes of the Human Genome Project and cloning in the late 90s, spurred politicians to act. In 2008, the United States passed the Genetic Information Non-Discrimination Act. This gave individuals protections from having their genome used against them in the workplace. However, despite these laws, there is one example of unapproved human DNA manipulation using this technology. In 2018, Chinese scientist He Jiankui was convicted of violating a government ban by carrying out his own experiments on human embryos, trying to give them protection against HIV. This unilateral action by an individual was widely condemned and resulted in prison terms for him and his accomplices. There are some broader ethical questions raised by CRISPR. The scientific focus remains on solving the problems that have always plagued humanity, like hunger and disease. CRISPR is a vital tool that has already shown promise in these areas, will be indispensable in addressing them in the future.